Hi students, welcome to the Chapter 9 lecture for Biology 112. This is Patterns of Inheritance. So this is going to be our introduction to the study of genetics. These are the learning outcomes that I will cover in this lecture. Gregor Mendel, which you've probably heard of him before, he's been nicknamed the father of modern genetics. Monohybrid crosses, which is primarily what Gregor Mendel was working with. Crosses between pea plants that differed in just one obvious characteristic. Alleles, which are variations of genes. So these would be the gene variants that would be responsible for giving a person blue eyes versus brown eyes, for example. Dominant versus recessive. This is basically how the alleles interact with one another. So, even though a lot of traits are what are known as polygenic, controlled by more than a single pair of alleles. You could say this one trait, such as brown eyes, is dominant to blue eyes, which would be recessive. I'll cover phenotype versus genotype. Phenotype is basically the appearance or the physiological functioning of the organism, whereas genotype is the actual genetic makeup that will determine what that phenotype is, to a large degree at least. Punnett squares. This is something you may remember from a high school biology class. It's basically a tool that you can use to predict the outcomes of certain crosses. Typically, these are just going to be monohybrid crosses that you're looking at Punnett squares with. Other patterns of inheritance, such as multiple alleles, and incomplete dominance. So these are things in addition to the simple dominant recessive inheritance patterns. I'll briefly talk about human and also some canine genetic disorders, sex-linked inheritance, and I will allow some time for Punnett square practice as well. Okay, so let's talk a little bit about Gregor Mendel. He was a monk who was living in what is now the Czech Republic. He worked in an abbey garden with pea plants in the mid-1800s. And he was really the first person to analyze patterns of inheritance. He did experimental crosses with pea plants to try to understand how traits were being passed down to offspring. And he was working in a time long before people understood the molecular basis of inheritance. This was long before people understood complementary base pairing, the fact that DNA is the actual basis of inheritance. But what he discovered still holds true to this day, and a lot of his studies are still used as the basis of our understanding of genetics. So there will be a lot of new vocabulary to learn with this lecture, but some of it's fairly intuitive. And some of it definitely is vocabulary that you've heard before. For example, traits. These are just varying characteristics in organisms. In the case of the pea pod plants that Gregor Mendel was studying, these would be things like different flower colors or different stem lengths. So these are some of the characteristic traits that Mendel was experimenting with. And what he was basically doing is crossing different varieties and seeing what traits the offspring had, and trying to determine why he was seeing certain patterns. Why do 75% of the offspring have inflated pods and 25% have constricted pods, for example? So Mendel experimented with plants to create both true breeding varieties and hybrids. Now, a true breeding variety, this is a variety that will breed true. This will be something that will predictably produce the characteristics that it has. For example, the purple-flowered pea plants are always going to produce purple-flowered pea plants. That would be a true breeding variety, whereas a hybrid would be the offspring of two different true breeding varieties. So the offspring of white-flowered pea plants and purple-flowered pea plants, for example. And it's important to keep in mind that hybrids is kind of a confusing term in biology sometimes. A lot of folks think of hybrids as crosses between two distinct species. So, for example, a cross between a wolf and a coyote. That's what a lot of people have in mind when they think of hybrid, is just two completely separate species breeding together and producing this offspring. But hybrid is also used to denote offspring of different varieties within a single species. For example, dogs. Always one of my favorite examples to bring up in biology, just because we've manipulated their genome so much through artificial selection. But a cross between a Labrador and a Golden Retriever can rightly be thought of as a hybrid as well, because those subpopulations, those breeds, have been separated for so long, and when you're bringing them back together, you are basically crossbreeding. You are introducing new blood into the offspring. So hybrids can also denote offspring that have resulted from just different varieties of the same species being crossed. So the nice thing about these pea plants is they can very easily be manipulated. So pea plants, like a lot of other plants, are capable of sexual reproduction. And in this case, you can transfer pollen from the stamens of a white flower to the carpel of the purple flower. So the carpel would be the female reproductive parts, stamen, male reproductive parts. And the pollinated carpel will mature into a pod, and then you can plant the seeds from that pod, and then you can get the offspring of that cross. Parental generation, or pea generation, would be the purple flowered plant and the white flowered plant. And then the offspring, also known as F1 or filial 1 generation, would result from that cross. And you'll notice here, what we have resulting is just all purple flowered pea plants. So this is one of the kind of quirks that Mendel noticed. Why is it that when I cross a purple flowered plant with a white flowered plant, that in the first generation, I'm always getting 100% purple flowered plants? What's happening to that white flower color, and why am I not seeing it? So Mendel performed many experiments. Most of these were monohybrid crosses, and this is just a cross between parent plants that differ in just one characteristic. So, for example, a monohybrid cross could be a cross between flowers with axial, or a cross between pea plants with axial flowers and a pea plant with terminal flowers. But all of these other characteristics would be the same. It could be a cross like we just saw between purple flowered plants and white flowered plants, but all of these other characteristics would be the same. That would be a monohybrid cross. So looking at that experiment again, pea generation, these are true breeding parents. So for example, if you crossed white with white, they would produce all white. If you cross purple with purple, they produce all purple. They're true breeding. If you cross those two different varieties together, the F1 generation, all of the plants have purple flowers. But then if you take the offspring, essentially the siblings from that cross, or from multiple crosses, if you took the F1 generation individuals, crossed those with one another, then Mendel would see this very characteristic pattern. He would see 3/4 of those F2 or filial 2 offspring would have purple flowers, and 25% or 1/4 would have white flowers. So he was very predictably getting this 3-to-1 ratio. And not surprisingly, he was very curious about why he was finding what he was finding. So he ran many trials with many different traits, not just flower color. For example, short or dwarf pea plants with tall pea plants. In the first generation, he would see all tall, and then in the second generation, he'd see that 3-to-1 ratio once again. From his results from multiple crosses with many different traits, Mendel generated 4 hypotheses. And remember that a hypothesis is simply an educated guess. It's something that must be testable. So the first one, there are alternative forms of genes, now called alleles. Number 2, each organism inherits 2 alleles for each trait, one from each parent. Number 3, if the 2 inherited alleles for a trait differ, one is dominant and one is recessive. And number 4, the gametes, what are equivalent to our sperm and egg cells, they carry only 1 allele for each inherited trait because the 2 alleles separate from each other during meiosis, and this is what's known as the law of segregation. So let's talk a little bit more about traits and where they're actually located within the chromosomes of DNA. This is the chromosomal theory of inheritance, and it states that genes are located at specific positions. These are known as loci, plural, or a locus would be singular form. And the behavior of chromosomes during meiosis and fertilization accounts for inheritance patterns. Now remember, we recently covered homologous chromosomes. Homologous chromosomes are those that we receive from each parent. We receive 23 pairs of homologous chromosomes from our parents. If we have kind of the standard human karyotype, homologous chromosomes have corresponding genetic sequences, but they may have different variations for each trait. So a locus would be a specific location on a chromosome that has a specific sequence of nitrogenous bases, and that in turn is going to influence what traits we actually have. So this would be a set of homologous chromosomes, one from a mother and one from a father, and you can see here that we have 3 different loci for these chromosomes. So locus number 1, we have a recessive allele, little b, received from the mother. From the father, we have a dominant allele, big B. For the second gene locus, we have little a from the mother and little a from the father. And for the third locus, we have big P, or a dominant allele from the mother, and the same from the father. So let's distinguish between genotype and phenotype. Phenotype is just the physical appearance of an organism, but I should also note that it's not necessarily traits that you can see. It has to be an actual manifestation of the genetics. So it could have something to do with physiology that we can't actually see as well. That is sometimes included in a more broad definition of phenotype. For most of the traits that we'll be talking about in this course, you can just think of it as physical appearance. For genotype, this would be the genetic makeup of an organism. The genetic makeup, of course, relies on the sequence of nitrogenous bases within the DNA molecule, within the chromosome. And in the case of genotypes, for our study of genetics, we'll really only be looking at a single locus at a time. So we'll be looking at the genotype at, say, the third locus on this particular chromosome. So a genotype is always going to be denoted by 2 letters, and those letters represent the alleles or the gene variants that you receive from each parent. So for example, little p, big P, little p, and big P, big P. Mendel's law of segregation states that pairs of alleles segregate or separate during gamete formation, during the formation of sperm and eggs, and then the fusion of gametes at fertilization creates allele pairs again. So what we have here is homologous chromosomes undergoing the process of meiosis after they create their sister chromatids. I'm going to pause here.